move on to our next speaker, Savannah Gibson. Savannah is a PhD student at Aston University in the Mycobacterial Research Group. Her PhD focuses on non-replicating persistent Mycobacterium tuberculosis by creating new in vitro models for studying this fascinating bacterial state.
Hi, thanks, Poppy. Thank you for giving me the opportunity to talk at this conference. As I said, I am looking into latent tuberculosis. So, as has previously been said, tuberculosis is a very well-known worldwide pathogen. It regularly infects around 10 million people per year, although these infections are beginning to come down now, and is very much indicated with multi-drug resistance. In 2015, the World Health Organization came together to form a worldwide collective on how to combat tuberculosis to try and aim towards its eradication. As part of this conference, they set out a load of milestones to hit by certain time points. On my bottom right, you can see the milestones for 2020. And now sitting in 2020, we have unfortunately missed all of them. They did a separate conference to discuss why we're missing these milestones, and several key areas were highlighted, areas like multi-drug resistance and tuberculosis, but especially latent tuberculosis. Current estimates believe that there is around 1.4 billion people. They are latently infected with tuberculosis throughout the world. So what is latent tuberculosis? Well, during the latent infection, the immune system comes together to almost package away in an attempt to control the Mycobacterium tuberculosis infection in these complex immune structures called granulomas. They try and center it in the center of these granulomas to starve them of essential nutrients, starve them of oxygen, and essentially make as hostile an environment as possible through macrophage releasing nitric oxide, pretty much no nutrition at all apart from cholesterol and very low pHs. These are all considered to be drivers of latent tuberculosis. And in varying in vitro models and in attempts to study latent tuberculosis, the general consensus is to use one of these drivers to push the bacteria into a latent state. Traditionally, people use hypoxia as the way to induce this latent state in TB, but other people have used states like nutrient starvation or a low pH. However, what we've been really focusing on is that these drivers don't exist in isolation. You don't have just hypoxia in a granuloma. You do have hypoxia in combination with nutrient deprivation, in combination with the only metabolite being cholesterol. So we tried to come together to form a sort of multi-stress model of latent tuberculosis that would hopefully be more accurate to the physiological condition. So we decided to see if we could form a model, two models really, one sort of as a control, which uses hypoxia as the only stress to induce bacteria into a latent state. And the one on my right is our multi-stress model where we have hypoxia in addition to a very minimal cholesterol-based medium. As you can see, these are very, very long assays. These have been going for 10,000 hours at this point. Now at the end of my PhD, these have reached 20,000 hours. And as you can see, these nice static growth curves are showing us that we have reached latent tuberculosis, and they remain viable, just not replicating and sitting and waiting for conditions to improve. The main trait of latent tuberculosis is widespread drug indifference. This has been very widely recognized, and in hypoxic models, the first big headline really of latent tuberculosis was that in latent tuberculosis, it is resistant to frontline TB drugs, isoniazid and rifampicin. And in our hypoxic model, we got similar results. Recent years with widespread drug discovery and in clinical trials, we are seeing more and more drugs coming through that are active against latent tuberculosis, such as moxifloxacin, protamine, and bedaquiline. However, we repeated all of this testing first in our hypoxic model, which we saw complete similarity to other people's results. And then we repeated it in our new multi-stress model. and we lost all sensitivity across all the new drugs. We were resistant to isoniazid and rifampicin still, but also the ones that are currently in clinical trials, like protomanid, like bedaquiline. Going through a wide range of antimicrobial drug testing, we found one singular compound hit against our multi-stress model, which is we've called compound SG327. And this activity was very, very interesting, because normally we saw, whilst it wasn't very susceptible, quite a lot of compounds would hit against our hypoxic model, but this compound did not. It only showed activity in our multi-stress hypoxic cholesterol model. And here you can, on my top right, you can see this lovely dose-dependent response over time. These are over 1,000 hours assays to try and get a really broad look at latent tuberculosis, as it's such a long-reaching condition. So previous work on this compound library had highlighted that they were meant to be antifolate inhibitors. And particularly this compound library was focusing on DHPS, or FOLP1, and tuberculosis. So we tried to validate this as the drug target for this compound in tuberculosis. And to do this, we ran an overexpression assay, where you overexpress the target protein in your bacterium and then run the same sort of antibiotic susceptibility assays, with the basic principle being, if there is more drug target, you need more drug to exert the same effect. So the plasmid-only control showed the same susceptibility as we'd seen in our original MIC testing. And what was interesting is that tuberculosis has two DHPSs. It has one called FOLP1, which is the active version, and it has what is normally thought to be an inactive ortholog called FOLP2. Just to cover all of our bases, we overexpressed both in tuberculosis and ran the assay. And we were very surprised to find that FOLP1, the MIC remained the same. It remained exactly the same as we saw in the plasmid, but we saw the MIC shift we were looking for in FOLP2, in this inactive ortholog of the folate protein. This is very, very interesting. So we decided to dig into FOLP2 and see what it was doing. So potentially finding a novel drug target. The interesting detail is about FOLP2 is that previous research has shown that it cannot act as a DHPS. It's not functional, it's not a part of the folic acid pathway. It just looks very, very similar to FOLP1. So this was all very confusing, and we tried to figure out where it has to be doing something. If we can inhibit it, it's got to be having some activity in latent tuberculosis somehow. That activity only worked in a hypoxic and cholesterol environment, not in a hypoxic only. So the hypothesis we went forward with was that it has to be something to do with cholesterol catabolism under a hypoxia setting. And we went back and looked at the cholesterol catabolism pathway in a lot more detail and found around 8 steps in this pathway that involve oxygen to proceed. And obviously in your hypoxic environment, these steps are suspect. One of these steps is the hydroxylation of a cholesterol breakdown product, essentially from a phenol to a catechol, by 2 enzymes working in complex called HSA-A and HSA-B, the formula you can see on the screen. This hydroxylates the cholesterol breakdown product using oxygen and a reduced riboflavin. Interestingly, the research that found this mechanism showed that it was HSA-B that had this high oxygen demand. Obviously, in our hypoxic system, that really raised an eyebrow. And the next thing they'd shown was that HSA-A was considered essential for survival in a macrophage. However, HSA-B was not. So we moved forward with the idea that potentially what we're seeing here is FOLP2, instead of acting in the folic acid biosynthesis pathway, maybe it's providing an anaerobic bypass for this process instead. So to see if we were correct here, it's a bit of a stab in the dark. And we used FOLP2 in a biochemical assay to try and see if it could interact with riboflavin 5-phosphate. Instead of using 3-HSA, which is the cholesterol breakdown product, we used a substitute called 3,4-dimethylphenol. And we used a fixed concentration of riboflavin 5-phosphate, and then we purified some FOLP2 and just see if we saw anything. And very excitingly, we did. With a buffer, with a separate protein, there is absolutely no activity whatsoever. However, when all of these three conditions are met, we can see a lovely concentration-based activity of FOLP2 turning over this riboflavin. And then the next step was bringing it full circle, really. And if we're seeing activity with a riboflavin in this scenario, can we inhibit it with our original inhibitor? And the answer was yes, we could. At a high concentration of our original compound, we found we could completely knock out the activity of FOLP2 by turning over this riboflavin. And yeah, we are moving on with this activity. We're going to try and, this is all very preliminary data, and we're going to try and shape this up a little bit, but unfortunately my time in the lab has come to an end, so someone else will be picking up this project. And yeah, is there any questions? Many thanks to Aston University and my supervisor, Dr. Jonathan Cox.
Thank you very much, Savannah.